Across our planet, from rural villages to busy city streets, old diseases as well as new contagions will always lurk. From cholera, streptococcus, measles.
There's a lot of diseases where vaccines just haven't worked in the past.
From HIV, the West Nile virus, the Ebola virus, SARS.
Many of these diseases are in third world countries, they're in developing countries, they're in developed countries.
Many old diseases never die, and many new diseases are easily spread.
Diseases know no global boundaries. We have diseases across the world, and we have to be prepared to fight these diseases wherever they may exist.
Many old diseases can no longer be treated by the same old drugs.
Rise in resistance to antibiotics and the reemergence of a large number of infectious agents has meant that we don't have things under control.
How do we control a virus, a bacterium, a parasite? Pathogens that can create disease, tiny enemies invisible to the naked eye that can be carried by the air, by the water, by animals, by people.
It was absolutely essential to move aggressively in the development of biosciences here, to work on treatments for infectious diseases, detection of pathogens, and prevention of the spread of pathogens. These are critical for human health and for national security.
How do we build new vaccines? The new weapons that could protect millions of people in hundreds of countries. By building a new kind of institution. In 2007, the University of Maryland opened the Bioscience Research Building, a spectacular $69 million facility that is now home to the Maryland Pathogen Research Institute, also known as MPRI. What's so new about MPRI?
The Maryland Pathogen Research Institute brings together leaders in biosciences, computational sciences, engineering, and nanosciences. These collaborations within MPRI share the goal of developing a comprehensive program to diagnose, treat, and prevent the spread of pathogens.
The basic idea: draw together researchers from dozens of different sciences and then create collaborations, working alliances among faculty members from the University of Maryland and scientists from other institutions, including the U.S. Army Medical Research Institute for Infectious Diseases, the National Institute of Standards and Technology, the U.S. Food and Drug Administration, the National Institutes of Health, and the U.S. Department of Agriculture.
We're actually going to try to incorporate the teaching.
The true benefit of MPRI to our faculty on campus is really the synergy which can be developed between researchers in different disciplines.
In the war against pathogens, synergy is the secret weapon.
You know, we see it every day. I told someone recently that I can just walk down the hallway, get a cup of coffee, and I come back smarter because of the interactions that I have with my great young colleagues.
Synergy can come from a coffee break or from a complex interdisciplinary collaboration between an organic chemist and a microbiologist. A collaboration to invent a new vaccine, or perhaps a better vaccine, one that's free of the dangerous side effects caused by many current vaccines.
If you get a big response on your shoulder from where they injected you, you're not going to want to take the vaccine. So we were trying to think about how to detoxify vaccines, how to make them more acceptable to the population, less toxicity.
A less toxic vaccine could be used to inoculate children from meningitis or protect soldiers from anthrax.
We got together and we said, well, let's redesign our thinking. How could we make it a better product? How could we make a vaccine that would actually work?
Start by enlisting a microbiologist and an organic chemist.
The synthetic chemistry turns out to be quite complicated and hard, and I've certainly sort of left the complex biology to some extent to Dan.
Both of us understood our half of the project, but neither of us could do the other half of the project.
Then train them on the state-of-the-art tools of advanced biotechnology. And finally, fire up the secret weapon, synergy.
And it turns out that the vaccine that Phil's built that we then tested works amazingly well. And so if you think about revolutionizing healthcare, we have a new platform for how to make a vaccine, how to deliver a vaccine.
At MPRI, synergy works. And it works with difficult diseases. Cystic fibrosis, for example, can clog the lungs with infections, leading to poor breathing and eventual death. And one of the pathogens that causes those lung infections is Pseudomonas aeruginosa, the research target for two different kinds of scientists, a computer scientist and a cell biologist.
I know about sequencing technology and I know about computational methods for analyzing DNA sequences. I don't know that much about Pseudomonas aeruginosa. Vince knows a huge amount about Pseudomonas aeruginosa.
Having the ability to rapidly go through the strains that have similar characteristics from the clinical side allows you to quickly focus in on the genes that may participate in those types of infections.
When you have the sequence, the genome sequence of a bacterium for the first time, that allows you to get a very detailed look at what it is that makes this bug tick.
What makes this bug tick? How does this human pathogen launch its attack? And how does a human cell respond? The answers can only come from basic research, but research with practical payoffs.
What we'd like to do with these genome sequences is start to identify new targets, both for vaccines and for new antibiotics.
Many diseases that attack the lungs are airborne, and some are so infectious they can only be studied in a high-containment biosafety lab, and so deadly that they are potential weapons in biological warfare.
Pathogen that most people don't know is Francisella tularensis, and it causes a disease called Tularemia. It's been the target of bioweapons programs, both in the United States and Russia in the 60s and 70s, and now it's a major concern. These are organisms that the CDC, the federal government, Department of Homeland Security, have decided are of both utmost importance to study, but also to be secured because they're dangerous. Because of that, you have to work with this organism in a specific type of containment facility.
At MPRI, researchers use a biosafety level 3 lab to study a number of pathogens that are potentially lethal if released. Dr. Kevin McKeever works on the Tularemia pathogen, and Dr. Brenda Fredrickson studies another dangerous and invasive pathogen, the West Nile virus.
It's found in every state in the continental US as well as Mexico and Canada. It doesn't typically cause disease in Africa where it's endemic. The reason that we study these pathways is with the idea that we can somehow develop inhibitors of the virus. So we can use a cell's own defenses to stimulate an immune response to the virus.
Once again, basic research can have big payoffs, and not just for understanding the West Nile virus.
When I look at a pathway that inhibits West Nile replication, if I can develop a molecule that will trigger that pathway, I will not only block West Nile replication, but I may block other RNA virus, such as HIV, such as measles virus.
Out of a disease like West Nile, perhaps a new way can be found to fight an old disease like measles. That's not serendipity, that's synergy at work.
These are all things that have come out of MPRI, which would not happen at most research institutes because they're not built that way.
If we take the best brains, connect them with high-end biotech tools, and focus their research on real-world health problems, the payoffs can be profound. Not just deeper understandings of disease pathogens, but new vaccines, new products, and new technologies that can deliver them to a world at risk.
There's no question that the rest of the world is taking note of MPRI and what we're doing here. People are starting to understand that MPRI is something that's going to be very important in the future. It's going to be the next big thing.